Hello everyone. Let's get started. Today we're going to be talking about microbial growth. By definition, when we talk about the growth of microbes like bacteria, we're not talking about the cell itself getting bigger and bigger. We're talking about the cell dividing and creating new cells. So we define microbial growth as an increase in the number of cells in a particular population. Now, the process most commonly used by bacteria to divide and thus grow is called binary fission. During binary fission, a single bacterial cell, what we sometimes refer to as a parent cell, will go through this process of division and create two genetically identical daughter cells. Recall that these daughter cells will be clones of the parent cell because they will be genetically identical. Binary fission is a form of asexual reproduction. It's basically one organism copying itself, creating two new copies of itself. Now, the time that it takes for one parent bacterial cell to become two new daughter cells is referred to as the generation time. And we shorthand generation time with a lowercase t. The process of binary fission is actually quite complex and involves a number of different proteins. But overall, the process involves a duplication of all of the materials that are inside the parent cell, including the DNA molecule, and a process referred to as elongation, as those materials literally increase the size of the cell. Then there is the development of essentially a wall in the center of the cell, referred to as a septum. And finally, the membrane of the parent cell will pinch inwards and then eventually break apart, producing the two daughter cells. You can see these things happening in the drawing over on the right-hand side of the slide. There is a parent cell depicted at the top, a rod-shaped bacterial cell. The first thing that's going to happen in binary fission is this elongation step. And they have drawn it particularly exaggerated here. But the point is important, that everything that is inside this parent cell, all of the materials that are inside that cell, including the DNA molecule, any materials that are dissolved in the cytoplasm, all of the ribosomes, everything that's inside that cell has to be duplicated. That's going to cause that parent cell to increase in size. Next, there has to be a septum formed to help divide that cell. And that septum literally forms directly in the center of that elongated parent cell. And as that septum matures, it will cause the membrane to literally split between these two brand new daughter cells. There will be a full set of cellular material over here in this daughter, including a complete copy of the DNA. There will be an identical complete set of materials in this daughter as well. When bacterial cells divide or grow, they create clones of themselves. They create daughter cells that are genetically identical to them. In the time it takes for one parent cell to become two daughter cells, we refer to that as a generation, or the generation time. Each of those daughter cells will then go through another generation time and create two daughter cells of their own. So across time, as each generation time passes, we will exponentially increase the number of cells that are being produced from that one parent cell. If you look at the image on this slide, on the right-hand side, you can see a spread plate. Remember, a spread plate is produced when we put a certain volume of broth culture onto an agar dish and we spread it out evenly across the surface, allowing cells to land across the entire surface of the plate. We then incubate that plate and allow each cell to go through binary fission again and again and again and again, producing an individual colony. So you can see that there are many, many, many individual colonies growing on this agar plate. And each one of those started with a single cell. Remember, we also said it is possible for two cells to land sort of on top of each other on an agar plate and grow what looks like a single colony when it's really two colonies. But for the most part, when we spread out cells and we get a whole bunch of individual colonies that all look identical, each colony came from a single parent cell. Each colony contains millions of identical daughter cells. And you might say to yourself, how is that possible within a normal incubation period of about 24 hours? Well, the generation time for many species of bacteria is relatively short, in the area of 20 minutes or so. And because this is an exponential type of growth, within 24 hours, we've had enough generations to pass, enough generation times have passed, that we get into the millions of cells in each one of those colonies. And of course, that's the only reason we can see those colonies, is because there are enough cells there that we can visualize them. Now, binary fission is certainly the most common way that bacteria grow. Most archaea also use binary fission, but they use different proteins to do it. In other words, they're doing the same general process. They're going through elongation, and then the development of a septum, and then the splitting apart of the parent cell. But the actual proteins that are involved in that process inside the cell, the actual enzymes, for example, are different. The other thing to remember is that archaea are capable of other forms of division beyond binary fission. The archaea, for example, can also use budding, like yeast, and a process called fragmentation, depending on the species. While I've described binary fission as being a very simple process, it's not. It's actually a very complex process. It has many, many steps in it. And each step involves different proteins, including different enzymes that each have a particular function to accomplish. In fact, inside the cell, there has to be the construction of a piece of material called a divisome in order for binary fission to proceed. This structure, this divisome, is actually what will create the septum, or the wall, that will be used to guide the division of the two daughter cells, one from the other. If you translate that word divisome, it means dividing body, and it's actually a collection of different proteins that come together and work as a single unit, not only to build this septum, but to allow the septum to then contract, to get smaller and smaller and smaller, pulling the cell membrane in with it until it literally pinches the cell into two. The divisome is critical because it determines where the septum is built. And if you think about it, the septum has to be placed exactly in the middle of the parent cell so that each daughter cell is the same size. And so each daughter cell gets the correct materials inside of it. As you might guess, this process of binary fission requires energy. It's an energy dense procedure that the cell is going through when it divides. So let's look at binary fission in just a little bit more detail. Remember, the very first thing that has to happen is this process of elongation. The parent cell has to undergo a process where all of the materials are replicated, including the DNA molecule. The parent cell will physically elongate during this process. Now, that parent cell is eventually going to be divided in two. So there has to be a mechanism to ensure that each of the DNA molecules will be placed into each daughter cell. The DNA molecule will be copied during this elongation process. And then we need to deliver one copy of the DNA molecule to one end of the parent cell and one to the other end, so that when that cell divides in half, there will be a DNA molecule on each side. So those two copies of DNA, those two nucleoids, when they're first made and they're just sitting inside the parent cell, they actually prevent formation of this dividing wall, this septum, because they're in the way. So what you're looking at over on the right in this top diagram is a depiction of the parent cell entering into elongation. And again, literally elongating, literally getting bigger as materials are duplicated inside. The nucleoid that's inside the parent cell, that piece of DNA, is being copied. And the copy is being made right on top of the original molecule of DNA. Now, the way the cell is going to separate those two is by attaching them to the membrane. So one might be attached here, for example, one might be attached here. Now, when that parent cell starts to elongate and literally get longer in length, the nucleoids will be physically pulled apart because they are attached to the membrane. One will be pulled in this direction as the membrane moves this way. One will be moved out in this direction as the membrane grows in that direction. Eventually, the two nucleoids will be physically pulled apart from each other. And that's the point where it's possible for the divisome to build the septum. And that's what you're seeing down here in this part of the diagram. The divisome will be assembled in a space that opens up once the two nucleoids have been pulled apart. A ring of protein, a literal ring of protein, will be formed along the inside of that membrane, all the way around. Remember, this is a three-dimensional cell. So all the way around the inside of the membrane, a ring of protein will form, and the septum will be built out of that ring. The septum is simply a wall of material. And once the wall is in place, it starts to constrict. It starts to sort of shrink in on itself, eventually pulling the membrane in so far that it splits in two. Now, why do we care about all these details? Of course, scientists like to understand natural processes like binary fission. But there are practical reasons to understand all of this as well, and one of those is the targeting of new antibacterial drugs. If we understand which proteins are involved, if we know which individual protein is required for binary fission to happen, we can target those proteins with drugs. So, for example, new membrane has to be made during the elongation phase of binary fission. New cell wall has to be made in order for that parent cell to elongate. All of that is occurring as this septum is being built. Proteins are building this septum as well. We can target proteins with drugs. And if we can prevent the protein from performing its function, we can prevent the bacterial cell from dividing, and hence treat the patient who has a bacterial infection. Now, penicillins are probably the most famous example of the targeting of binary fission, because what penicillins do is interfere with new cell wall synthesis. If the parent cell can't build new cell wall as it splits into two new daughter cells, those daughter cells aren't going to have a complete ring of cell wall around them. And if they don't have a complete cell wall, they won't survive. So penicillins work so beautifully, so elegantly, because they interfere with the production of cell wall. You and I don't have cell walls around our cells, so the penicillin won't bother us. It will only bother bacteria that are trying to survive and divide in our body. Now, obviously, penicillin is no longer useful against all bacterial infections. In fact, it never was. But we're always looking for new antibacterial drugs. And the more we know about processes like binary fission, the better we're able to investigate and try to find drugs that will target other steps in the process.